detail. What is high pressure liquid chromatography? HPLC stands for high pressure liquid chromatography, or sometimes high performance liquid chromatography. So it is a one type of liquid chromatography where the mobile phase is liquid and the stationary phase is solid. So it is solid liquid chromatography, and even there, even we have liquid chromatographies also. Stationary phase is very highly non-volatile liquids. There are few cases where we have such kind of things. So why we should use when there is a liquid chromatography? See, paper chromatography, whatever we have discussed now, it is a liquid chromatography. Then why I should do high pressure liquid chromatography? This high pressure liquid chromatography is very expensive. It is that's what it is not available in every college. Only few rich institute can offer this one. So why we should go for HPLC, high pressure liquid chromatography, or high performance liquid chromatography? Generally, when you for your routine chromatography techniques, it takes a lot of time to separate the molecules, and we don't have much resolution. See, in case of paper chromatography, that whatever you have separated, that carotenes, xanthophylls, chlorophylls, they are having almost, they are overlapping. They don't have much demarcation for them. That can be achieved by using your high pressure liquid chromatography. The demarcation process is called as resolution. In microscopy also we use the word resolution. Making 2 points clear from one another is called as resolution. In paper chromatography, they are overlapping. That I don't want that part. So even in microscopy, if these 2 points are overlapping, the image is going to be blurred. If these 2 points are separated, it will be a clear image. Hope you understood. And one is to make a better resolution. Second, to make the process fast. It is a normal chromatography, like this pigments. If you want to separate plant pigments, that's the entire process you can wind up within 5 minutes, and with an excellent resolution you can do here. So that is the main objective of HPLC. Here the system is concerned, you need to develop high pressure. How to develop a high pressure over there? Here you will be having a packed column. If full packed column will be there, and we will be having the pumps where you can pump the solvents into it with high pressure, high speed. Like if you are having a fountain kind of pumping, then there is a possibility of high pressure development. You cannot do normal slowly. If it is moving, you cannot do it, that one. And this columns are generally these columns are very tightly packed, so that what happens here, the pressure can be created in this one. See, if you see, generally we'll be taking either single or two solvents in, and these solvents are generally degassed. Water is a big problem. Air bubbles are big problem in case of your chromatography techniques. So your degassers are attached, and after degassing, it will be, the solvents will be mixed together, and the pump, there will be a pump. It will be taking the solvents into the column. Generally, sometimes we'll be arranging the pre-column, or it is called as column guard. And this column guard helps if any impurities are there, and if any pressure leakages are there, or pressure increased pressures are due to kinking of the wires. A lot of pressure is built, and due to some knobs, if they are opened, the pressure is reduced. So this solvent will be transferred into the column. And before column, you will be having a flow splitter, and here sample injector. Whenever you are injecting the sample, the flow will be stopped, and after the sample is completed, then the flow is continue. Anyhow, I will show you this process. So this is basically HPLC system. And the chromatographies, everybody must be aware about this, but just I will do a little part of this. Partition chromatography, anyhow, I have explained with paper chromatography as well as TLC. Here mobile phase and stationary phases are partitioned into multiple floors or multiple zones. And adsorption chromatography, here adsorption chromatography, your compound is adsorbed to the solvent phase, the stationary phase. Your molecule is adsorbed, whereas rest of the things are removed away from that one. And one of the best example in adsorption chromatography is affinity chromatography also, that is there. Reverse phase chromatography. In reverse phase chromatography, the stationary phase is reverse phase. Reverse phase means hydrophobic. Normal phase is hydrophilic or polar, whereas reverse phase is non-polar or hydrophobic. And normally at industry level, all protein separation, biomolecule separation, we do with reverse phase chromatography. C18 column is a normal thing that we use in industry levels. Next, ion exchange chromatography. You must have studied anion exchange chromatography, cation exchange chromatography. Here, your stationary phase is attached with ions. Anions will be there, and it will be binding with the, it will be replaced with the other anions. And in the elution phase, you will be putting stronger anions in that one. Okay, this is ion exchange chromatography. Size exclusion chromatography, it will be like your agarose electrophoresis, or matrix will be there with the holes. So the molecules, see, some molecules of smaller size will enter into the matrix beads, and they will be eluted slow, they will be coming out slow, whereas the bigger molecules which are not going into the beads, they will through the bead, the sides of the beads, they will be percolating and they will be going fast. So what happened here, this is the case where the larger molecules will be eluted fast, whereas smaller molecules, as they are entering into the beads, they will be migrating or they will be eluted later. This is the difference in between the agarose or page to size exclusion chromatography. And affinity chromatography. Affinity chromatography, you know, it is a biological process where generally protein A, you must be aware, protein A is a staphylococcal protein. It binds with all type of immunoglobulins. Whenever you want to separate the immunoglobulins, so we'll be collecting the protein A and we'll be immobilizing in the stationary phase. So whatever the on the column, stationary phase that you are packing, that is attached with the protein A. And whenever you are putting the mixture containing immunoglobulins into it, so selectively this protein A will be binding with the antibodies, and rest of the thing gets washed, and we'll be adding highly ionic strength containing eluting buffer so that these antibodies also get separated. So what are the things that are required? Solvent is required, and solvent delivering system, that pump is required, injector is required, samples we need to pass, the column is required, and detectors, waste collectors, and recorder is required for this thing. This is a very old HPLC. Anyhow, you will be enjoying the new one. Very so this uses I have explained. Injectors. See, in case of GC, we are loading the sample with needle fix. You should be thorough in the loading. 1 microliter, 2 microliter. If you are not perfect in loading, what happened? Then there is a possibility of error. Whereas in HPLC, there will be a injection loop. Injection loop is there. So that injection loop, either you load more also, it will be taking only the fixed quantity of sample, and excess sample will be passed out. It will be thrown out into the wash basin. It will not be taking that one. So it is very easy to load into the sample into HPLC, whereas GC, that is there is some difficulty. And next is columns. Like as the techniques are concerned, we'll be using different. If you are separating non-polar compounds like amino acids, proteins, RNA, DNA, in such cases, even this caffeine, organic molecules, all biomolecules, whenever you are separating organic molecules, C18 hydrophobic column, reverse phase column is generally recommended. And whenever you are separating polar molecules, the molecules with charge, if you are separating, ion exchange chromatography is preferred. And whenever you are like you are looking to bind, when many things are there, you want only your molecule should be retained. In such cases, adsorption chromatography is preferred. And whenever you are doing like biomolecules, antibodies, if you want to separate, it is not only antibody, any antigen antibody based reactions, if you want to do separation of antigen based on the antibodies or antibodies based on the antigen, if you want to separate, then you can use affinity columns. But at industry, it is a very common to use the reverse phase chromatography. And this is the column. You can see column here. It is generally you will be having the columns of 3 centimeters to 30 centimeters. And these columns are two types: analytical columns and preparative columns. Analytical columns, we can load very little amount of sample. You can't load very high amount of sample, and its a job is only to separate molecules and give you the information whether your compound is present or not one. But again, you collecting back that substance may not be possible because very little amount of sample is being loaded over there. Whereas semi-preparative samples, semi-preparative columns, here you can load up to 5 ml. In case of analytical, hardly you can load 1 ml. Semi-preparative 5 ml you can load because it is a bit larger than that of analytical column. And here you can take out the fraction and you can do the characterization. Whereas preparative columns, you can load up to almost 200 ml. It is a very big column. And here generally whenever you are looking high quality products, high quality, I think for this COVID, we are using purified monoclonal antibodies, trimase or something, monoclonal antibodies against interleukin 6. And the cost, I think you must be aware, it is 60,000 rupees per vial. In such cases, high valued, in such cases, you require highly qualified, highly purified product. So in such cases, we will be using a preparative HPLC. Otherwise, normal chromatography techniques are quite enough for us. So for separation, I told you it is a early and good resolution and even recover the compound. There are certain things, column material, see here, they are called as shields. And if they are disturbed, column material is important, and how they are arranged, that is also important. And there's a coating material on the column, that what is there, that is also important. And temperature at which it is getting eluted is important. Flow rate, signal, and sample sensitivity detectors, these are the few important factors. And this already I have explained. Normal phase is a polar phase, reverse phase is a non-polar phase. Size exclusion chromatography is it is a just matrix like your agarose gel electrophoresis or page, but here the difference is molecules, if they are small molecules, will be entering into the beads, so they will be eluted later. Ion exchange based on the polar nature. Type of detectors, see we'll be loading the sample, and see in the chromatography phases, first equilibration phase is there. After equilibrium, see your column is made ready to bind with your molecule. Then you will be loading your sample. Then you will be having a wash, so that except your compound, everything gets separated. After wash, you will be doing elution, so that whatever your bound molecule is there, that also gets eluted. That is also going to come out. After this elution, your compound should be detected. It should be, its signal should be accessed by some detector. So these are the some detectors: UV visible detector and IR range absorbers, fluorescence, refractive index. Generally, refractive index will be using for crystalline compounds like sugars, and fluorescence if your molecule is having fluorescence capacity. IR and absorbance generally will be using for proteins and organic molecules will be using. And evaporated light scattered detector when your molecule is volatile, like methanol, such kind of things will be using this one. Generally, volatile compounds will be using in GC, but if it is less volatile or something you are separating by HPLC, we need to have this detector. And electrochemical, here in electrochemical, the charge of your molecule will be measured here. Mass spectrophotometric, here the separated molecules mass will be measured. Photodiode detector, this is absorbance based one. This is the basic of HPLC, because many of you may not be aware about the basics of HPLC with so I thought that you should be aware about the things. Now we'll do
High-performance liquid chromatography is a technique in analytical.
There is a just 3-4 minutes animation, then followed with there's a explanation about components and instrument. Then we'll be having an experiment.
Chemistry used to separate, identify, and quantify each component in a mixture. As well as other forms of chromatography, in HPLC there is a mobile phase, which forced by a pump to pass through the system. And there is a stationary phase called a column, which located in an oven where the temperature can be controlled. The sample can be automatically injected into an HPLC system by the use of HPLC autosampler, or can be manually introduced into the injector using a syringe. There is also a detector attached to the HPLC system, which measures the analytes after its separation in the column. The pump forces the mobile phase through the column, and then the detector, under high pressures. In the HPLC system, a vacuum pump and a degasser are connected to the pump and used to remove dissolved gases from the solvents. The pump drives each solvent to the mixing chamber, where mixing takes place under higher pressures. HPLC analysis commonly uses isocratic or gradient elution. In isocratic mode, the mobile phase composition remains constant throughout the procedure. Whereas in gradient mode, the mobile phase composition is changed during the separation process. Sample introduction can be accomplished in various ways. The simplest method is to use an injection valve. In the load position, the high-pressure eluting solvent flows to the column directly. The loop is loaded at the atmospheric pressure from a syringe via the needle port. Excess sample exits the loop via vent port. After loading the sample, the valve is switched to the inject position. The flow delivered by the pump flows through the loop, forcing the sample ahead of it, flowing to the column. Then, the valve returns to the load position, and the mobile phase moves the sample through the column. The separation is based on differential partitioning of the sample components between the mobile and stationary phases. The component which has more affinity to the mobile phase, consequently less affinity to the stationary phase, travels faster and eluted out first. And the component which has more affinity to the stationary phase, consequently more interaction, travels slower and eluted later. This separation can be carried out according to which type of HPLC used. There are two main types of HPLC: reverse phase and normal phase. Reversed phase has a non-polar stationary phase and moderately polar mobile phase. One common stationary phase is a silica which has been modified by attaching a straight-chain alkyl group to its surface, such as the octadecyl group, C18, or the octyl group, C8. In this case, the more hydrophobic the analytes are, the more retained they will be on the stationary phase. The more polar they are, the more they will prefer the mobile phase. In normal phase chromatography, the stationary phase is polar, and the mobile phase is nonpolar. This method separates analytes based on their affinity for a polar stationary surface, such as silica. In this case, the more polar the analytes are, the more retained they will be on the stationary phase. The more hydrophobic they are, the more they will prefer the mobile phase. As compounds elute from the column, they interact with the detector. Different types of detectors can be used, such as the UV-vis detector, which showing an absorption spectrum in the ultraviolet or visible region. For UV detection, a deuterium discharge lamp as a light source is used. And for components detection in visible region, a tungsten lamp is used. In UV-vis detector we can also find entrance slit, lens, prism or diffraction grating, exit slit, flow cell, and detector for absorption measurements. Light from the lamp is shown onto the prism and dispersed according to wavelength. When the measurement is performed with a specific wavelength, the angle of the prism is adjusted so that the light of this wave can shine on the flow cell. As the compounds elute from the column, they enter the flow cell, where the bonding and non-bonding electrons of these compounds can absorb energy in the form of ultraviolet or visible light. The manner in which the final data is displayed is based on the computer and software. The number of peaks present can indicate how many components are in the mixture. Usually, the x-axis of the HPLC chromatogram shows the amount of time taken for the analytes to pass through the column and reach the detector. Typically, the y-axis, or the area of the peak, is a reflection of the amount of a specific analyte that's present.
Yeah, now I will explain you different components of HPLC. One particular Agilent instrument is here, and I will explain you first different components and how it works. The software, actually nowadays all instruments are supported with softwares. So the operation we cannot. It should be automated operation of HPLC. So I will demonstrate you first this one. Then we'll go with one example run so that it will be easy for you. So first, see, the solvents are kept over here. This A and B, two solvents are there. Along with one, there is one more bottle which is kept for wash and maintenance of column. Generally, before running, the columns are kept under a solvent, appropriate solvent. Methanol is a more appropriate appropriate solvent to preserve the columns. See the C18, C8, C10, C12. These columns are hydrophobic chain is there in this one. So that should be maintained intact. If it is a dry, what happened? The hydrocarbon chain, or not only this reverse phase column, any column that gets spoiled, the internal matrix get deteriorated if you are not maintaining it appropriately. So you should maintain, after your process is over, you wash the column thoroughly, and you wash the pipeline tubings, everything, and you load your whatever the preservation sample, and you can so that sample, what a preservation sample maintenance it is called as washing and maintenance. This is the third bottle. Yeah, this is the solvents. These are the solvents, and these are the pumps, and this is a vacuum degasser. This is the auto sampler here, and this is the column compartment. I will explain you in detail. These are the detectors. We can have the multiple detectors, and these are the solvent bottles. As I told you, generally we'll be using two, and one more bottle is a maintenance bottle. That is for running, washing the pumps, washing the tubing, and maintaining the column. You can bypass the remaining things and you can maintain. And here you are having a degasser. This is a degasser. This is a vacuum pump, and this is a sampler. See, nowadays we are getting auto samplers, but you can do manually also. Here here you will be having the tubings that I will explain you. And this is the column chamber. This is the column chamber, and here whatever your sampler is there, that is connected to this one. And this is the detector site. This goes to the detector. Here in the column, separation takes place. The sample comes from here and goes out into the detector. And detectors could be multiple. In most of the time, whenever you are doing for the proteins, you will be having a UV detector. This DAT detector is a UV detector. And fluorescent detector also we can use if you are separating specific biomolecules like cancer markers or any antigen antibody binded things, if you want to screen such kind of things. But as per your observations, so we'll start first you need to start everything. Here only one detector, UV detector, is on because fluorescence work, and it should be operated through your software. So you need to open your software, and I told you this is a washing. One is a washing solution. So we'll wash first immediately as you start the instrument. It will be flushing with high speed. Normally, if it is a 1 ml per minute, it will be doing with 5 minutes. Now you need to create a method, you need to develop a method, you need to decide what should be the flow rate and what should be the, see here two solvents are there, water and methanol. So we are taking 50%, and this is the purge valve. Whenever you keep high speed, what happened, whatever the bubbles or any residues are there in that one, they will be washed out. This is general maintenance. So immediately first beginning you should do the purge. Purge is nothing but high speed flow for a shorter while, generally 5 minutes or 10 minutes will be keeping. And even after the purge, if something is remaining, if the air bubbles are remaining, generally they will be very small, so don't worry about them. Then you close the purge valve, you are closing. Now you design, the purge should be closed. Now you design the method for your experiment. And before that, as I told you, you open the column and make sure that there are no leakages, and you remove whatever the column content is there in that one. Because when you do the purge, the liquid does not flow through the column, because high pressure, if you are passing through the column may get damaged. So will not be passing through the, that is bypassing the column. So you will be opening the site of the column. And one more thing, the kinkings are very important in case of HPLC, because so many wirings are there. If it is kinking, you know that it develops a lot of pressure, and if it is pressure develops, then you cannot do. So where you need to select a method, otherwise you need to develop your own method. Here we'll develop a new method. New method will see. So some name should be given whenever, you know that whenever you are creating a file, there should be a name for it. So here lab sub to, you need to differentiate from the others. So lab demo, some name has been given. And we'll go with the automated one, not manual. For manual, you need to sit over there and every step you need to follow. And here the flow rate you need to decide, normally 1 ml per minute. Here methyl and methanol, we are going with 75%. Generally here, whenever you are designing a method, B concentration only will be given. So based on that, A will be automatically calculated. And in the presentation, you must have seen isochromatic separation and gradient separation. In isochromatic separation, throughout the elution process, see the loading and washing does not matter for us. Elution is most important. In elution, if you are using the same concentration of A and B, otherwise if even one, for the entire process, if you are using only one concentration, suppose here B is 75%, A is 25%. If you are using the same thing for the entire process, that is called as isochromatic. Generally, sugars are separated by this method, whereas proteins are separated by gradient method. In gradient method, what we'll do, will initially see, generally B will be acetonitride. In the for when we separate the proteins, B will be the acetonitride, mostly acetonitride with some phosphoric acid or some other substances, little amount. So whenever you are doing this, initially you will be using B concentration 10%, then 20%, then 30%. What happens here, when you use the 20%, weakly bound proteins, shortly bound, whatever the proteins are there, they will be eluted fast. Then see, as the more hydrophobicity, as the bigger the protein, more the hydrophobicity, it will be binding tightly with your C18 columns. So it won't be easily separated with lesser concentration of that is acetonitride. As you increase the concentration, so here our job is to separate only specific our protein. You need to wash the non-specific binded proteins. So in this case, the B concentration gradually we are increasing, and at one particular concentration, maybe 70-80% of acetonitride, you will be eluting, you will be detaching your protein bound to the C18, and that is coming out and entering into the detector. So that is called as gradient. That you should be able to differentiate in between the isochromatic and gradient separations. So here we'll be taking for your just demo purpose. So we are mentioning isochromatic. And here the pressure is a very important thing. If there is a leakage, if some there's a knobs, first it is called as, if some furs or knobs wires, if they are detached, then what happen? Pressure suddenly shoots down. If there is a leak column, there is a wire going into the column. If it is coming out, then what happened? The pressures will be going down. And sometimes if there is a kinking, the flow is not going at all, and there is a kinking or the bend of the wire. In such cases, what happens? The pressure will be shooting very high. These both the issues are dangerous for HPLC. Going down pressure, so you should monitor the pressure. You should fix some pressure. Generally, high pressure you can keep whatever size, but lower pressure you should go around like it should be less than 100. The 60, 60, 70 we should keep the pressure, so that if if there is a removal of wires or something, then immediately we can do the things. And the other side, you can see here time and AB concentration. If you want to create a gradient, and during the elution, I'm telling you clear, not during the washings, only during the elutions, we'll fix different time intervals, 1 to 2 minutes. Let's be 75, be 20%, and then 5 to 10 minutes be even you can design that for 5 minutes B concentration should go from 40 to 60. By minute wise it will increase the concentration, like you must have seen the gradient PCR, like how the temperature is increased, and here also will be increasing the gradient of B concentration in this case. So if you want to construct the gradient, you can do here also. So the pressure 70 and 40 is taken, and detector we are selecting, that is a UV detector DAT selector. That is the one we are supposed to select for the proteins. And what you want, like total height, area, because quantitation will be done based on the area. And how you want the report outcome, output report, how you, because it is a soft copy, it will be giving what you are expecting that it is coming. And what you want to measure online, A concentration I want, a B concentration, flow rate, and temperature we can measure. And this is after data getting, what do you want to do the analysis? That is also, if you can fix, then it will do for you. Now detector, that is a detector, and you can set the wavelength also. Normally for proteins we'll be going for 220, but this is organic molecule, so 254 has been selected. And save it in a particular whatever the method that you have developed, it should be saved. The same name lab demo we are giving. Now system is see here, these are the sample loaders. This is a Agilent is a very this particular system is an advanced system. So this is a auto sampler tray, and where you will be putting the sample 2 ml vial this is 2 ml vial, and with a septum, so that it will penetrate, it will pierce from the top, septum will be pierced, and this is see 2 ml, and the sample volume should be minimum 0.5 ml, and it could be up to 2 ml, but normally you can go with 1 ml, that is very good, and it will be taking very little amount of sample. But this is the auto sampler, so you can take, and this is sequence. You are supposed to give the information where your sampling bottle has been kept. It cannot do by its own. So the position has been given over there, and after giving the position, and we'll be asking it to start. First we'll be saving the sequence, so that subsequently also we can do in the same thing. Lab demo will be saving. Now we'll be asking run sequence. We'll be asking it to start in the sequence. Now then the HPLC will be continued. 1 second. Yeah, here once you start switching, this is to explain you the components. and how to do HPLC. Okay, in the next part, see, first I explained you the principal component, and then we explained you different components of HPLC, even software, how to handle, how to start, how to demonstrate, how to design a method for running a pump, and isocratic and gradient methods, and UV selection, detector selection, then sequence selection, then immediately then you should start the experiment, so that it will be running the HPLC and giving you automatically, it will be giving you the, once it is started, once the sample enters into the column from the loop, it starts into the mainstream, it's the HPLC will be started. Then after the once the HPLC started, you need not to do that, software will be collecting the data, it will be analyzing for you, and it will complete areas, everything will be like a printed form, it will be giving you everything. Okay, now we'll go with one example of doing estimation by HPLC.
Within complex solutions, one of the main components needed to run HPLCs are mobile phases. This is the different sets of mobile phases set up for HPLC that you can use to run your samples. And this is one of the type of HPLCs that we have here that we use with the mobile phases to accurately run your sample. To run HPLC, you need mobile phases. Shown here are different kind of mobile phases. What is a mobile phase? A mobile phase is basically a solution that runs through a stationary phase called a column. These are the two different kinds of column used in HPLC to run your mobile phases through. To run your mobile phases through these columns, it is recommended that you filter your solution. When running a solution such as buffer through these columns, it is highly recommended that you filter your solution. Now we'll show you how to filter your mobile phases. To start the filtration process, for the setup, you will need mobile phase, funnel, sidearm, trap, tubing, vacuum pump, member filters. You can either use 0.45 UM or 0.22 UM. To initiate this process, you will select a membrane filter, insert it on the membrane filter holder, then tightly secure the funnel on top of it. Then you will have your mobile phase on the side, ready to be poured. The next step is you turn on the vacuum pump, and make sure there's no bubbles in the tubings. Then you slowly pour your mobile phase in the funnel. It is being filtered in the side flex. Now you wait for the filtration process to be completed. What is not shown here is sonicator water vat, which is recommended when you use a buffer. The sonicator vat will decrease your mobile phase and can cause any contamination. The other main component here is your trap. The trap takes in any bubbles, any contaminations that can flow back in the funnel through the tubing. Once you see that there's no mobile phase being filtered in the side arm anymore, now you can turn off the vacuum pump. Now open the funnel. Now that you have your filter mobile phase, take your filter mobile phase and pour it in a clean container that is ready for filter. Attach it to the appropriate mobile phase tubing line. And make sure your tubing lines are flushed into a waste container so it does not carry any carryovers that can cause contamination to your system. The next step is to purge your HPLC. To purge your HPLC, you will use the purge valve by opening it, which will be turning it to the left. Now you'll see that the tubing will eliminate any bubbles that could be in the mobile phase tubing. Once there are no bubbles flowing through the mobile phase tubing, your purge process has been completed and you can close your purge valve now. Now we will talk about the selection of the column and attaching it to the ferrule. This is called a ferrule. For this setup, you will need a PEEK tubing, cutter, column, and ferrule on each end. You'll take your PEEK tubing. To cut it appropriately, you insert it in the cutter. Instead of cutting it, you spin it. Now snap the PEEK tubing in the site it was cut at. This gives you a nice round shape that you can insert in the ferrule. You take this end of the PEEK tubing, insert it in the ferrule, just about an inch from this side. Take the ferrule and you attach it to the column. To know which side you will attach this ferrule to is by knowing the flow of the solution in the column. The column has a point indicator which way the solution flows. In this case, the solution is entered from the pump, from the HPLC from this side, so you attach the ferrule by tightly securing it on this end. Once the flow of solution comes in, then it comes out from this end of the ferrule out to the detector for it to be analyzed. Now this column is ready to be installed in the HPLC system. Once your system is adequately primed, your column is in place, you have chosen the proper mobile phases, your system is ready to be equilibrated. When running your standards, you want to make sure you run your standards in multiple injections. That will ensure repeatability. There are many different types of HPLC systems. When operating one, important factors to keep in mind are selection of proper mobile phases, understanding the procedure in use so you can follow it accurately, and selection of proper instrument parts such as column. These will ensure a successful HPLC run. Thank you for watching.
Yeah. Actually, sample run, that's acquiring data that is explained with GC. So almost both acquiring data is same for HPLC and GC. So I thought it will be repeated. So